our TA has created a separate email address for you to send, for local students to send your homework submissions to. So this will be only for receiving homework from you guys, so you can be sure not to miss any emails. So please note the new email to use for Yahoo. All right. So today we're going to pick back up over here with our first set of R code, bioconductor code for exploring some genomic data. Last time I introduced you to bioconductor, and we ran this in order to get a function that we could then call that would download data and functions for us. And then we ran this, which installed a particular package called Biomart. Make sure this is available to us here. I'm going to run this over in R. You should be able to do what I'm doing right now, yes. Okay, so what is this Biomart thing? Biomart is its own, it's a standalone suite of software. Biomart.org. And primarily we're going to use this, so as we're interacting with genomic data, there are various repositories online where you can go get data. So if you want the sequence for gene X, you can go to a repository and get it. If you want the list of proteins that are involved in a particular pathway, you can go to a repository and get it. So we're going to use a handful of these repositories. There's not just one, unfortunately. The first one we're going to see is called Biomart. And I was going to show you this page. If you Google Biomart bioconductor, then here is some documentation on how you can access Biomart using the bioconductor stuff. So you might see a page like this repeatedly throughout the semester. This is at bioconductor.org, where again you have lots of packages that have bioinformatic applications. And if you click on a package, you'll see a page like this. So this is a description page for this package. It's the interface to Biomart databases. And inside the Biomart database system, there are multiple repositories that you can access. And I'll talk a little bit more about this one at least, because this is the one we are going to use most. It's called Ensemble. Okay. And you've got some citations about it, and down below, let me just mention this, by the way. They're with an R package. You download R and you get all this functionality that comes with the base installation. Right now, we want some extra functionality that didn't come with R, so we're downloading a package. It's a bioconductor package. There's a whole bunch of packages that aren't bioconductor. Bioconductor is just R packages that are specific to bioinformatics. For R packages, if you want to learn how to use them, so you read somebody's paper in the bioinformatics journal, and they have a neat method, and it looks like they've implemented their method in R, and they've provided a package. So you need to figure out how to use their package. There will be at least one, but usually a couple of different ways to learn about it. There is, in this case, a user's guide, and I'm going to open that up in just a second. There will always be a reference manual, and I would not recommend this as your first stop. The reference manual is an exhaustive listing of all the functions that are in the package. So in the BioMart package, there is a function called attribute pages. There's a function called export FASTA, and so on. Lots of functions. For any one of these, so I just scrolled down to the first entry. This is the first function that's in this package. It's called attribute pages, and it gives a summary of the attribute pages. And there will usually be pretty limited description of what each function does. So it's not, the easiest way to learn how to use a package is not to open up the reference manual and just read about each of the functions individually. The user's guide, if there is one, there isn't always one available, which makes it hard to get started, is more helpful. These are also helpful. So you can create, if you create an R package, you can create what's called a vignette. And a vignette is a PDF document that walks through the use of the package. So I won't go into it. I'll let you look at it sometime on your own if you're interested, or if you want to do this process of learning about a package. But it's a PDF that has example code in it. Suppose you want to download the sequence for gene X from the Ensemble database. There might be a little section in the PDF that says, here's the specific code that you can call from BioMart package to do that. And you can click the code to actually run it. So a vignette is kind of a handheld tutorial of how to use the package. It's a good place to start to learn. Let's look at the user guide just for a moment. It's another PDF that the authors provided. And kind of similar to a vignette, it walks through some example tasks. I'm just going to look at the first couple of sections just as an example of what an R package is all about. So give an introduction and tells us that the BioMart package, it's all about an interface to databases containing genomic data, such as the Ensemble database. How do you get started? You get started by selecting the database you want data from. Are you interested in data on humans, for example? Or are you interested in the Ensemble database or one of these other ones? So there's a PRIDE database, which is for protein data. We're going to look at Ensemble. So pick which database you want to look at. Then, and I'll show you this in our code in a second, you can list all of the databases that are available. Pick one, and you tell R, this is the one I want to use. So we're going to use this useMart function, and that creates a connection to, in this case, the Ensemble database. Now I can communicate with the Ensemble database through BioMart. I can also see within that database what specific data sets are available. List data sets for the Ensemble database, prints out this, and basically this is saying for a bunch of different species, we can access genomic information. One of which will be Homo sapiens, so you and me, but lots of other ones. There's cow, there's mouse, there's rabbit, there's all kinds of things in here that you can get genomic data for, sequences of genes and such. So over in our R code, we're going to make a connection to Ensemble. We're going to get some information about the human genome in just a moment. Once I pick which database I want, I use a particular function to tell R that. And then it's a question of how do you extract the data that you want. What data do you want? To see what variables are available to you. So think of this Ensemble database for Homo sapiens as a big data set. And there's different variables in the data set. What variables are there? That tells me what kind of information I can get out. If I list the quote filters, that tells me what I have available to me. So in the ensemble database for Homo sapiens, there are these things for a variety, for a whole bunch of different genomic locations. So think DNA locations. Take human DNA, stretch it out, get millions and millions of those A's, C's, T's, and G's, and in order to get a particular sequence, say, I will want to be able to say which chromosome do I want the sequence from. Is it chromosome 1 or chromosome 2 or dot, dot, dot. Where on the chromosome is my gene located, maybe? If I know chromosome 12 in between bases 1,014,000 and 1,380,000. That collection of A's, C's, T's, and G's, that's exactly what I want. I can specify that. So things like that. What pieces of information do I want about that region? I can get the available things to specify with this list attributes. So do I want, for example, a description of the gene that lives in that region? Or do I just want an identifier for that gene? Okay, so we talked last time about DNA, and we said that if you zoom in on one particular region of the DNA, there might be the A's, C's, T's, and G's in that particular region, and that constitutes a, quote, gene. And we said a gene, roughly speaking for our purposes, is the A's, C's, T's, and G's that when you read them and make a copy of them and then turn them into something else, you get a protein that does a specific function. So for all the genes that we know about, 20,000 something genes that are known to produce proteins in humans, how do you name them? What is, for example, I'll show you a specific gene in a minute in the context of breast cancer. There are specific genes, specific places in the DNA that are known to be relevant to breast cancer. When this gene maybe gets a mutation, it turns off. It doesn't express anymore, and you get cells that don't die, cancer. Or when that gene gets mutated, it turns way up, it's way more active than it's supposed to be, and you get disease in some other way. So these genes that are known like that, they have names. So that I don't have to say, I'm talking about the gene on chromosome 13 located between 1,014,000 and 1,044,000 or whatever. In the context of breast cancer, there's a gene that's given a symbol, BRCA1. It's a symbol. There's different types of symbols, depending on who you ask, they might have a different name for the same gene. So we can get out some of these names from these databases. So let's say BRCA1 is a gene. It shows up a lot in the literature because it's known to be involved in some important processes. I'd like to make a connection to this ensemble database for humans, and I want to download the sequence, the A's, C's, T's, and G's for gene ID BRCA1, for example. The main workhorse is this function here, getVM, which we'll use in just a second, and that's how I would actually get that information. Make a connection to ensemble, specify the human database, getVM with ensemble gene ID BRCA1, say, would give me A's, C's, T's, and G's for that particular gene. Or it could give me something else that I'll talk about soon. Okay, so let's go actually run some code then. I think we're ready. Library. Go ahead and save this. Has anyone tried to run this? Did you try to run it? There's no R on these computers? Yes. works on your home computer. Okay, I'm going to work with the tech folks, the IT folks, to get these computers set up. So hopefully, yours has R. Did you maybe look under all programs? Okay. I'd be surprised if R was not on these computers. All right, so I'm just waiting on this thing to install the Biomart package real quick. And then let's do something. Let's use the ensemble data set, and let's look at the human data set within ensemble. So I'm running a couple of commands that we just saw in that user's guide. I'm just going to do this. The first two commands here, they just print stuff out. So tell me what databases there are available to me, and within the ensemble database, tell me what data sets there are specifically. We saw the output of those two things in that PDF we just had open. So I'm going to create an object called Mart and call it whatever I want. It's just a variable that we're creating, and it is a connection to that specific data set. And I need to library it in. When you do this BioC-Lite Biomart, it downloads all the code that defines the thing, but you don't have access to what's inside of it yet. You've installed it, but each time you want to use it, whenever you open R next time, if you want to use the actual functions, you have to load them into that current instance of R. And you do that with the library command. That's library. Now, I can call functions that Biomart has. So let's create this connection, Mart. Error, space required after the public identifier. Let me look at the documentation for useMart. Okay, so I need to specify a Biomart database. If I list marts, that should tell me what databases there are. Okay? System public. Is anybody else getting this error? I didn't get it on my computer. Personal library, you think? Okay, let's try this again and see if it will ask me if I want to create a local library. Personal library instead. On your personal machine, probably won't have this issue. Some of these machines in the classrooms, I'm not an administrator on this computer, for example. Let me just read through what we're about to do. We'll wait on the R to finish doing what it's doing. We are going to extract the gene symbols and ensembles' own identifiers for all of the human genes. These are for known regions that code for something in the genome. So these aren't all things that code for proteins. If you think of a gene as a particular location in the DNA, if you make a copy of that and send it off to another part of the cell, it will be turned into a protein, and the protein is what goes and does stuff. We'll talk a little more as we go along, and another thing I meant to mention was I posted a couple of links to Khan Academy videos. So if you're brand new to all this genetic stuff, I'll just reiterate that you don't have to be a biologist or a geneticist to do this class, but the more you know about what DNA is and genes and proteins and that central dogma, it'll help you out. I linked to 2 lecture videos on Khan Academy that talk about that stuff. And so one little extra detail that we'll deal with repeatedly is that your first thought about a gene, think of it as a piece of DNA that will code for or, quote, express itself in terms of a protein that goes and does something. So there's a gene that will code for it's time to destroy this cell because it has gone through its life cycle. We need new cells, let's kill this cell. So I would turn on, or the body would turn on that gene, and it would express itself in proteins whose job it is to go actually physically kill the cell. Okay. But more and more lately, it's becoming apparent that there's other functions of DNA. So there would be other pieces of DNA that don't result in proteins, but still do something. They code for something that is involved, that is important to the life of the organism. If you watch the second video that I posted Khan Academy for, it mentions this word epigenetics. And so it's good to have at least heard that word. If you do research with people who are doing bioinformatics on campus, for example, chances are they're going to say, can you help me out with these epigenetic data? What does that mean? Epigenetics refers to ways that DNA can be used to regulate what's going on at the molecular level, but not necessarily by coding for proteins. So for example, here's 30 genes that are involved in the breast cancer process. Some of them code for proteins that physically do things that result in protection from cancer or the actual cancer process. Other pieces that we're talking about, pieces of DNA, maybe we don't call them genes, but they are active locations. They are active sequences. What they might do is create another molecule, maybe an RNA molecule, that goes around and turns on or turns off other genes, or turns up or turns down other genes. They regulate the expression of other genes. So they are not just empty, they have a role to play. Even though they don't turn into a protein, there is active locations, lots of active locations in that do things, not necessarily with proteins. So that's referred to as epigenetics. That comes out from the early thoughts that all there is to DNA is the genes that code for proteins. And if that's all we look at, there's a lot of the DNA that we just ignore, because a lot of the DNA does not code for any known proteins. So there was jargon junk DNA used to indicate DNA for which we don't know what it does. So we don't even worry about that DNA. Let's just focus on the genes. But more and more it's becoming apparent that some of that, quote, junk DNA is actually very important because it does this regulatory stuff. One example of this regulatory thing that is interesting and kind of illustrates how complex an organism can be, 20,000 something protein coding genes. That's a lot, but there's billions of people in the world and we all look different. And you can't quite explain the complexity of life with 20,000 different proteins. But you can explain it whenever you bring in this epigenetic stuff, that I could have the same gene as you, but I have different other supporting regulatory things that make my gene act differently than yours. Even if we have the same sequence for that gene, mine might express differently than yours because of this stuff. So one example that I've used in the past is Ecoli, which is an organism that if depending on what circumstance it finds itself in, it does different things molecularly. And I can't remember it's lactose and glucose, two sources of sugar. If there is glucose in the environment that the E. coli is living currently, it has the ability to express certain genes, turn on certain proteins that specifically degrade and process the glucose for energy. If it's in an environment where there is no glucose, but maybe there's only lactose, it will turn off its functions that go after glucose, and it will express a different suite of functionality that eats lactose. That's epigenetics. That's the ability of an organism to, even though its DNA did not change under those two circumstances, what the DNA is doing changes. All right, let's see how R is doing here. Okay. And let's see if we can library this in. And let's see if we can run this now. Okay, so I'm going to have to get our tech folks to help us out. I'll work on that today. All right, so let's do the best we can in the meantime. When we call this code down here, I am going to ask for, in that data set, Homo sapiens, in the ensemble set of data, please give me all the what are called H, G, and C symbols, and give me all of the ensemble gene IDs. These are two different ways to identify or name the gene. I'll show you H, G, and C in just a minute. It's a collection of unique symbols for each gene, like BRCA1, those letters, that's a H, G, and C symbol. That says everybody knows which gene we're talking about when we say that. But if you go to another database like Ensemble, Ensemble doesn't call it BRCA1. It has its own unique code. So if you're doing research and BRCA1 is the gene you're studying, you might want to have all the symbols that you need in order to get data off of the repositories. So let's use Biomark to get, for every gene in the human genome, these symbols. That's a big object because there's lots of things that just came out. In R, if you have a big object, like a big matrix, 50,000 rows by 30 columns or something, if you just type the name of the matrix, it will kill your display. You know, it'll just scroll. But if you say head, your matrix name, it'll just print the first few rows. So in terms of just taking a peek at big stuff in R, this head function is helpful. All right. What we're going to get out is, like I was saying, both protein coding genes as well as some genes that are known to be more epigenetic in nature. They don't code for protein, maybe they code for RNA. That would be for regulating the expression of other things. It will also give us information on so-called pseudo genes. And what a pseudo gene is, let's look that up for us. Is a location in the DNA that is thought to have been an evolutionary hand-me-down from ancestors, but which, due to, say, mutation, has lost its functionality. For example, this inset here, here's one particular spot in the chimpanzee genome, GTG ... Here's the corresponding spot in you and me, and it looks almost identical. So where the chimp has a G, we have a G, we have a T, T, G, G, and so on. But there are some differences. So there's what's called an insertion somewhere between chimpanzee and you and me. There was a mutation event which resulted in one extra nucleotide letter being inserted into our DNA. And similarly, there was a mutation event that resulted in one letter being dropped from our DNA. And there was a mutation where the T that came from the chimpanzee changed to a C. So if there's a mutation, think about what that means. I have my DNA, it's my A, C's, T's, and G's, but I smoke lots of cigarettes over the course of my life, which creates damage. It can create damage to your DNA. Damage to your DNA can look like mutation. DNA breaks, comes back together somehow, and and something was deleted in that process. Or it breaks, and there's other nucleotides floating around, and one or more new ones jumped in where they shouldn't have been. Or instead of when we were making copies of our DNA, when our cells were replicating, instead of the T that was supposed to come here, a C was mistakenly placed. And then if this propagates for me, all my cells start looking like that, then when I have children, I will pass that stuff on to them. So there could be a mutation that then carries forward through generations. So there's a lot of complication, a lot of depth to these genomic data, and we'll see some of that when we use this biomart functionality. Because what I'm going to do is ask for all the genes for humans, and some of them will have symbols that are recognizable, like BRCA1. I'll show you another one in a second, the Mike gene. But a bunch of them will not have known function because they are, say, a pseudo gene, and some of them will be this epigenetic role. All right, so I'm limited in what I can show you with biomart today. I'll get that fixed as soon as I can. I was going to, when we did this and print out the results, we would see for all the different genes that are known for humans, their symbols. So suppose there was a specific gene that we wanted to study, like BRCA1, or here's another one called Mike. Let me go over here real quick before we do that. So HGNC has its own website, genenames.org. If I come here and I can ask for information on any old gene I want. So I'm going to say, tell me about BRCA1. Enter. Here's all the articles and resources that have relevance to that. The first one is what I want. Some of these others are related to BRCA1, but here's the main entry for BRCA1. So you can learn about what's known about a particular gene. This particular gene has lots of stuff that we could dig into. For example, there'd be citations about what we know about it and its role, in this case, in cancer. There would be the actual sequence for that gene. So here under this section, nucleotide sequences, I'm going to click on one of these. There's a bunch of different databases and resources. GenBank is one that's widely used. Click on GenBank. And then I get specific information about where this gene is located and what its sequence is. I'm going to scroll down to the bottom, and here's the actual sequence for the BRCA1 gene. So this tells me if I were to go to somewhere in here, it should tell me what chromosome and what specific region of that chromosome we are at. If you went to that location and started reading, these would be the letters you would actually see. So you can see these aren't small. Genes aren't necessarily small. This is a few thousand letters. So when the cell makes a copy of that specific spot and takes that specific code to the manufacturing warehouse, it turns it into the BRCA1 protein. There's also a different sequence. So this here, these are not A's, C's, T's, and G's. These are, when we talk about proteins, they are also can be thought of as a sequence of letters for our purposes, but those are not A's, C's, T's, and G's. They're not the nucleotides that are put together. They're now what are called amino acids that are put together. So a protein is called a polypeptide, which is a sequence of amino acids. Those are just different molecules put together. And what happens when you put those together in a linear way is that they fold in on each other into a three-dimensional structure. And now you have this protein. Three-dimensional structure is actually relevant to the function of the protein. You have a virus floating around in your cell. The protein that is designed to kill that virus will need to be chemically able to connect with that virus and also physically able to connect with that virus. So think of these. Zoom way in on your cell, there's physical little things floating around, and they have structure, and they also have charge for chemical bonds. So the protein and its physical conformation needs to, for example, be able to come and sit on the virus and physically attach to it. When people design pharmaceuticals, they need something that will go to a specific place in the body, maybe, to fight a tumor, and they need to be able to latch on to the right piece. And so part of the design of a pharmaceutical drug is this chemistry of which amino acids do we need to put together, bearing in mind how those are going to end up into a three-dimensional structure. And is that consistent with what the target looks like. All right, so we know what a gene is. We know that genes are made up of these A's, C's, T's, and G's. For any given gene, there is a sequence that defines that gene. You go to one chromosome and start at this place and start reading all the way to that place, and you'll see this. That information, that's the encyclopedia or reference manual that gets copied, carried over to the manufacturing plant, and as we'll talk about, when you read these letters, take that A, G, C, if you read these letters in chunks of 3, they code for amino acids. So A, G, C, AGC, look it up in here. Yeah, okay. So here's AGC. If you get a triplet as you're reading along your sequence that's to be copied and turned into protein, if the first 3 letters you see are AGC, then what will happen is the amino acid called serine will be brought in and put into position. The next 3 letters were TCG, and U, we'll talk about this, but U can be interchangeable with T if we're talking about RNA. So here, I'll say this is what we would point to. This is a different, it's the same amino acid. So different codons can code for the same amino acid. So what would happen is we read these things off, or the cellular pieces read these things off in triplets and turns the triplets into these amino acids. So that's how we end up with a sequence of these letters. Each letter is an amino acid. There's serine right there. There's alanine right next to it, and so on. This is the protein that does stuff. There is an Ensemble website as well. I'll just show it to you here. I come to Ensemble, and let's ask for BRCA1 again, see what it gives us. Let's take the human gene BRCA1. Largely complementary information from what we saw at H, G, and C. So if you end up dealing with data like this, it just is it's the nature of the beast that you have to be able to work with these different databases, communicate between them and stuff like that. So the Ensemble page told us BRCA1 is on chromosome 17. It's located between these, in this region. So that means go to the 43,044,295th letter, start reading, and go through there. And if we went back here, that's what we would read. If we started there and started reading, that's what we would see. Ensemble also gives us neat pictures that the biology collaborator would make more sense of. Here's a representation of the chromosome up here, and these colored regions correspond to that region where the gene lives. And an interesting tidbit from looking at this picture is all of these colored spots are coming from this region called BRCA1, but there's a bunch of them. And what that means is if, for example, you go to the BRCA1 location, but through regulation, epigenetics, you only transcribe, express this subregion. So here's BRCA1, and for whatever reason, we're only going to express code, put into action a subset of it. You get a different function. You don't get the same BRCA1 gene that you would get if you turned on the whole region. So this is yet another way that there's complexity among organisms. 20,000 something protein coding genes for humans, but there's all these other locations that code for other things. And even within one gene, you can turn it into multiple proteins. We'll talk about this a little more, but maybe you've heard the jargon splicing and alternative splicing, alternative transcripts. One gene can give rise to multiple functions. It can get complicated. Almost out of time for today, but the next thing we're going to do is look at peptide sequences for one specific gene, the Mike gene. I'll talk about it next time. It's just another gene that is important and known to have a big role in certain disease processes. We, up here, got all the gene symbols. And now for a particular gene, let's get the sequence. Because we're doing statistics, and I don't want to have to come over to Ensemble or to HGNC and type the sequence in. I want to be able to read this stuff in to R so that you can actually do stuff with it. And here's one way you can do that. You can use the get sequence function to get either the A, C's, T's, and G's, or in this example, I'm going to get the amino acid sequence. A protein is called a polypeptide. All that means is it's peptides put together, and all a peptide is amino acids stuck together. So when I say peptide sequence, what I really mean is this, amino acid sequence. So when we run that code next time, it's going to give us this amino acid sequence for this particular gene. When we do that, we will see evidence of this issue, the fact that, okay, you asked for the Mike gene and the amino acid sequence for that, and what we'll actually get is it'll get a list of amino acid sequences. Here's one amino acid sequence that is known to arise from the Mike gene. Here's another one, here's another one, here's another one. Depending on how you make a copy of it, you get different protein products. We'll see that. One last, I'll get to that next time, but well, just so we don't need to spend too much time on it. One last minor detail has to do with how this goes from turning the copy of the A, C's, T's, and G's into the actual protein. So think about a gene. A gene is located maybe in this region. So think of this line up here as a piece of the DNA. Lots of A, C's, T's, and G's. And the BRCA1 gene lives right there. So if you start here and go all the way over to there, that defines BRCA1. In order to create the BRCA1 protein, or one of them, not all of this that is in between those two places is needed. So there are what are called introns and exons. Exons are refer to the regions in that sequence that actually are required to turn into amino acids that define the protein. Everything else is referred to as an intron. It's a non-coding region. So the body needs BRCA1 protein. What's going to happen is it goes to the corresponding spot in the DNA and it makes a copy of that whole chunk. Now there's a little piece, a small, relatively small strand of A, C's, T's, and G's. Actually, the T is replaced detail floating around. But I don't need all of this because these are introns. And then what happens is that enzymes and other things come in and chop it up and stick together just the pieces that we need. This is the coding piece, the A's, C's, T's, and G's that are relevant for the amino acids. And that's what's then turned into the protein. That process of chopping and sticking back together is called splicing, RNA splicing. You take DNA, make a copy. The copy is RNA, which is its own single-stranded piece that's free to move out of the nucleus. Before it can be processed into a protein, it has to be, quote, spliced and turned into this. The way these kinds of things happen is by, let's make a copy of that BRCA1 gene, and now depending on specifically what we need, or the body needs, we can chop it up different ways. So if I want this particular function, I only need this sub-region of the gene. If I want this function, I need that region and I need that region. So there are alternative ways to splice a gene. Alternative splicing, which is another mechanism for complexity in humans. All that to say, when we come back here and say, give me the sequence for Mike, we're going to get lots of sequences. And they each correspond to something slightly different. We can then look those up. Let's say the Mike gene, the third transcript that's known. Let's go look up what that does. It's involved in such and such. So why do we care about doing this kind of thing? And we haven't done any statistics. Well, what we're going to do eventually is we're going to have this P by N matrix of numbers. Each row corresponds to a feature. Might be a gene. Gene 1 is Mike, maybe. And then for each of N individuals, I have a number. I have 10 cancer people, 10 healthy people. And I have a number for each person for Mike. The bigger the number, the more copies of Mike is floating around in that sample. The more active Mike is in that sample. And so then we're going to do statistics, because I have 10 numbers for Mike in healthy, 10 numbers for Mike in cancer. Let's do a t-test, say, and ask, is the mean expression level of Mike different between cancer and normal? So we will do that for all of our P features, which will be thousands and thousands and thousands, which means we have thousands and thousands of P values. And a typical task would be to filter out from those thousands a small subset of features that look strongly different between the conditions. Those are the ones that we would potentially learn about the biology from. So here's my list of 100 genes that had really, really small p-values. I take them to the biologist collaborator, and the next question is, what are those genes? So I might want to do this kind of thing. For this, gene 421 had a p-value of 1 times 10 to the minus 14. Looks obviously different between cancer and normal. What is that gene? I can look up its symbol. I can ask Ensemble for details on what is known about it. I can get its sequence. I can get its amino acid sequence. I can find out all the different transcripts of it. So think of the bioinformatics that we're going to be doing as kind of an exploratory task. We have these massive data sets, very rich, expensive data sets, and we want to filter out from them the nuggets of information that tell us about what's going on molecularly. So a lot of it will be search among a haystack of features to find a handful of interesting ones, and then go connect that list to known biological stuff. So that, for example, we found these 100 genes. We didn't know anything about cancer before this study. We did this study. We found 100 genes. And if you look up what those genes do on Ensemble or one of these databases, lo and behold, these genes are involved in cell cycle termination. They are involved in telling the cell when to die. So if you were to break those genes, turn them off or turn them up or do something unusual to them, you're going to you're going to throw a monkey wrench into the process of the cell dying. So we just learned probably cancer involves the cells aren't dying when they're supposed to. Okay, we're out of time. We'll pick back up on Monday and holler on the discussion boards if you have questions about homework, but you hopefully will be able to run this code without issue on your personal computers. I've had issues pretty much every year I've done this, trying to get the classroom computers to work right. But on my personal laptop, I don't have these issues. Okay? Good weekend.